Good morning, and thank you very much for the invitation to participate in the conference. Analytical methodology and toolkit for improvement of quality attributes and process optimization of biopharmaceuticals. Over the past 10 years, drug discovery efforts have been progressively focused on protein research with improved knowledge about complex functions in cells. Now we need to reflect all those changes and adjust our approaches to study proteins in isolated form. And we talk a lot about replacement of old inefficient drug discovery models with new. I would like to give you some examples and establish a bridge between old models and how we need to approach now studies with proteins in isolated form, because when we go back to 2005, about 30% of the best selling drugs were small molecule drugs screened over G protein coupled receptors. Now, 10 years later, we have about 50% of the best selling drugs proteins, biologics. So we need to think in different way. And the main difference between the old model single drug target interactions, now we want to think in more complex way. Independent signal activation triggers in cells kinases, ion channels, less studied redox properties of proteins, and in coordinated physiological responses. On the other side, oxygen consumption is changed, increase of oxidant reactive oxygen species, and accordingly increase amount of antioxidants to keep the cellular homeostasis and keep cells alive. So I want to focus today my talk about redox properties of proteins, because when we work with proteins in isolated form, the main concerns of researchers properties related to oxidation, then abnormalities related to aggregation, oxidation, and low activity. In terms of thiol-based redox homeostasis, in the past, we studied a few compounds such as glutathione and thioredoxin with reverse conversion of disulfide bonds in such kind of compounds. So now we have a lot of statistical data. They illustrate that almost every single protein has such kind of reversible conversions of disulfide bonds. And this is example in human proteome. We have over 400,000 cysteine residues existing, about 5 to 20% oxidized in form of disulfide bonds. And from crystal structures, we identified almost 20 possible disulfide bond configurations. These are 3 examples of bonds with higher potential to be incorporated in redox reactions. So this is just illustration that we need to consider all those types of redox properties of any single protein in the cells, and I will give you some examples. In terms of reactive oxygen species and metal-induced oxidation and degradation, in the past we studied a lot reactive oxygen species generation in cells in response to stress, and some mechanisms of action have been proposed, such as phantom reaction in presence of iron and copper. Now we want to link those data with other sets of data related to disulfide bond rearrangement, and this relates to protein-protein interactions or rearrangement of motifs inside the cells, and in case of disulfide bonds, they are protected from zinc finger motifs. Then we need to think about correlation between metals in cells and how they should affect protein behavior in vivo and in vitro. Antibodies, real data, whether they are used as drugs or for research diagnostics, very often they give multiple bands on gels, and instead of single band corresponding to intact subunit, you can see oligomers or disintegrated subunits. So we need to analyze this when we start projects working with proteins in isolated form. Also, surprisingly, in the both subunits, you can see in crystal structures different types of oligomers formation. So those type of statistics now help us to better build our understanding about heterogeneity and abnormalities such as found in our daily studies with antibodies. Also, metal content. This is totally new area of research, and we need to pay more attention about the existence of metals in antibodies. Let's say in this particular research, zinc ions interact with histidines and promote antibody receptor interactions, but in many cases they could be harmful, and this should be one of the reasons for degradation and oxidation. In the past, we studied antibodies assuming variable and conserved regions, and in this slide is illustration about cysteines. Usually we ignore cysteines for research, assuming that they have irreversible disulfide bonds. Recent studies shows interconversion in this red connecting lanes, and in case of IgG4, for example, such kind of interconversion may explain disintegration and existence of bonds corresponding to the half subunit. Paradigm shifts and great illustration for new models and proposed mechanisms of action for many proteins studied before, and now with much more statistics from crystal structures and other data from other proteomics research, we can build better models and understanding. In the case of adenylate kinase, it was studied quite in the past as model peptide, because there is a large opening of binding domains in the core during the catalytic reactions. So now we have a lot of new data For example, in this lead domain, there were found extensions with zinc finger motifs. In the past, we believed that zinc fingers are sites of interactions with DNA, and now a lot of new data shows significant contribution of these domains in enzymes, and we try to explain now what is the function of these domains. So the power of bioinformatics to align sequences, let's say here they aligned sequences of several family members of the enzyme. We mapped their substrate binding domains. In this part is the extension with the zinc finger. And what is surprising, this zinc finger belongs to a family member from prokaryotic cells, but in human, there is lack of this extension. On the other side, zinc binds to the interface, and this is one of the reasons why we can explain trimer structure here, or zinc in the substrate binding domain replacing magnesium. So this type of data, when we analyze in parallel, provides a powerful strategy to think about, consider some new properties and design experiments in more efficient way. So going to the analytical methodology and toolkits, how we should study proteins for research, diagnostics, and drug discovery, differently than the sequential way, what was applied for many years. Now we need to study in parallel properties and develop methodology based on bioinformatics, specific for each particular sequence and system, and considering factors and instruments applying to analyze these factors in parallel. So whether they should be applied on a single, multiple, or routine-based assays sequence, and how to find interactions, this should be matter of choice for researchers, depending what kind of instruments they have available. And I would like to give you some examples about those type of approaches when we start with data mining upfront and apply analytical and quality during the whole process of research and development pathway in order to get more consistent results. In case of cell culture and upstream process, when we express proteins in foreign cells, normally factors we consider cell type, strengths, gene constructs, pH, temperature, metabolites, and want to distinguish process from product-related factors in order to better manage these projects. Now, based on the new data coming from different fields of life sciences, we want to consider also factors such as oxidants, oxygen consumption, media composition, sequence and structures, and again carefully to distinguish which factor relates to process, which one to products, in order to get good activity and stability in the end of the process, because this is most important criteria how we consistently get results in the research. And in terms of yield, this is usually main goal at the beginning. We should consider yield as a stress factor for the cells of expression. So in terms of higher yield, this should be related to high stress, high oxygen consumption, and reactive oxygen species. Accordingly, much higher potential to have degradations as a result of this. So linking all this data is very helpful to manage the process. In terms of recovery and purification, normally we think about purification, chromatography resins, buffer composition, and more attention should be paid in future for additives, how to replace native environment with synthetic in order to keep the proteins in functional state as much as possible close to the native. In terms of pH variations and redox properties, oxygen consumption and reactive oxygen species, metal-induced oxidation. So all this data deriving from the previous studies, we need to transfer and carefully analyze in the next steps. Again, activity and stability should be monitored during every single step of the purification process in order to analyze which parts of the small molecules bound to the protein could be explained in the course of this purification process. The big advantage of design of experiment approach is where we study more than two factors in parallel, and search for correlation between them is to get more precise information with fewer experiments and integrate all the studies and data in one. Analytical and quality should follow all the steps in the process. So examples of cost-effective assays, they could be applied on a daily basis, absorption spectra of purified proteins. This is something every single laboratory can do. For example, the blue curve represents pure proteins, and if we apply this kind of spectra for many other steps of purification and analysis, we can get much more information. For example, the gray curve represents existence of aggregates, the blue curve bound small molecules, and even you can estimate coordination of some transition metals such as copper, iron, or cobalt, and shifts in the wavelength may indicate local arrangement of the aromatic residues in the proteins. In other words, those kind of techniques could be used to study a lot of properties of the proteins, in addition to kinetics and molecular weight, what we studied in past. We can add more information related to oligomers aggregates formation, conformational changes, binding affinity, stability, small molecules coordination, and also oxidation and metal content, either as spectral assays or plate-based assays. So as you see, in general, we can take full advantage of instruments in laboratory to start analyzing proteins from the very beginning of their expression and purification process. And this should help to comprehensively analyze proteins with multidisciplinary design of experiments approaches. Bioinformatics should be the first step of this process in order to find differences between cells, type cells of expression, original cells, what we use to study proteins, and searching for multiple factors and correlation in order to get more information and consistent results. This in general allows improvement of quality and a reducing of production costs. Thank you very much, and I'm open for questions.
Short the time, so if there's any questions, so okay.
So do you get to study the affinity of these drugs that you produce? So do you use techniques like MST, ITC to study how strong these drugs interact with the target proteins?
My personal experience, you mean?
In general.
In general, yeah. I went through a large variety of proteins used for research, drug discovery, and diagnostics from different origin, and actually all those concepts I illustrate right now, they're a result of my own research, and in my collaboration with colleagues from University of Toronto, Canada, Europe, and Japan, we developed, let's say, this is share of information and common strategies for the importance of cysteine research in our daily work, because for example, one of my studies as postdoc in University of Toronto chemistry department, there are great research groups working on model bacterial proteins. So many of those concepts we took from proteins. They are model proteins in biochemistry, bioorganic research groups, nothing related to market valued proteins. But on the other side, they give great ideas how you can manage projects related to other types of proteins. Let's say I had also experience with G protein couple receptors, and all those concepts I just mentioned applied wonderful to these model proteins, because they are membrane, and usually people assume that membrane proteins are totally different than soluble proteins. It's true. On the other side, there are features we need to apply, let's say, when you perform these multiple sequence alignments and search for correlation between sequence and structure conservation. We didn't have very many structures for G protein couple receptors because for membrane proteins. But now when you compare structures, sequence alignments, it becomes very obvious where we should put more efforts in our further research to identify critical factors. Let's say in GPCRs and other signal proteins, one of the features is this crosstalk between cysteines. And this is totally new area of research. So again, we need to develop different understanding about proteins and how they are functioning in cells, and all those proposed mechanisms of action based on few crystal structures don't work anymore because we play with statistics these days. It's non-realistic to develop proposed mechanisms of action based on 2-3 crystal structures, what was practiced before. So what I want to say is that we are excited to live in new era of research when a lot of new data coming and make possible to go to the different level of understanding how proteins work and how we can maximize our potential to study proteins in our lab on a daily basis.
Questions? Oh, okay. It's my pleasure to felicitate the certificate of your excellent
Thank
presentation.
you.
And
Thank you.